Good morning, everybody. Good morning. Can you all hear me and see the screen properly and clearly? Everybody, just tell me on the chat box. Come on, everybody. Is the screen absolutely perfectly visible to each one of you? And am I audible to everybody in the class? Just tell me yes or no on the chat box, and then we will move to today's session on metabolism and the enzymes first part. Yes, Fatima, Anandu, Sai, everybody, very good morning to everyone. Yes, so today we are going to discuss about two different important topic, that is metabolism and enzyme. So this is particular thing which we are expecting a question to be asked for your NEET examination. Okay, everybody. So let's start. Those who are watching the session, don't forget to hit the like button, do share it with your friends, and subscribe the amazing platform for more updates on biology. Okay, everybody. This is our amazing Telegram group at t.me/neat_biopoint where daily session updates, regular discussion, polls, and quizzes takes place. So those who have not become a part of our small family of Biopoint, don't forget to click the link in the description box and become a part of our group. So let's get started with today's topic, that is none other than metabolism. Ready, everybody? Give me hearts on the screen and let's move forward with the super amazing class. Guys, we will be having tomorrow also, will be most probably tomorrow or day after tomorrow, it depends. We'll be having one more lecture on this chapter, biomolecules. Okay, we'll be having just one more lecture on metabolism, and after that we will simultaneously go with two different things. That is, we'll be starting for CBSE students, we'll be starting the term 1 revision as well. State board students can also join for the same, because we will be focusing on NEET and we will be moving forward, because for term 1 CBSE students also they have to do the MCQs. Yes, Calvin, how is your health going? Good morning. Yes, how is your preparations going? Just 40 days more for your entrance examination, which we all were awaiting, right? And CBSE students, the date was yesterday, your results, but till now they have not officially confirmed or posted anything regarding the results. So let's hope for the best, and we are expecting it to be released today afternoon at any time now, at any time in next 2 or 3 days. Starting from today, 10th standard students will definitely get the results. So today we are going to start the metabolism. Okay, so listen very carefully, stay tuned till the end, everybody, and clear your doubts as well. So, metabolism. What is actually metabolism? Right from the chapter Living World onwards, we are studying that metabolism is actually, in certain cases, in most of the cases, it is a characteristic property of what? It is a characteristic property of living organism, but not in all cases, but in few cases. Okay, everybody. Yes. So let's discuss what is metabolism. All biochemical reactions which are taking place inside the body. Okay, all the chemical reactions which are taking place inside a living body or a living system constitute metabolism. Clear? So we have already discussed what is metabolism in our chapter 1 itself. So guys, today at 7 p.m. you are having a test on Living World. So don't forget to attend. We'll be posting it exactly in the same model as the NEET. Okay. So don't forget to attend it. So metabolism are of two types. Everybody listen carefully. Metabolism are of two types: catabolism and anabolism. Catabolism and anabolism. Okay, catabolism and anabolism. Catabolism means it is a destructive process. That is, it will break down the process, the metabolism which leads to the breakdown of complex substances into simple substances. Anabolism means it is a building up. Anabolism means it is a building up of materials. Okay, so there are so many examples which we will come across in this chapter, and we will learn in detail about those in the plant physiology and human physiology chapters. Okay, so I'll not be explaining too much about the graphs and processes which are shown over here. Okay, so guys, removal of carbon dioxide from amino acid to form amine is an example for a metabolism. Removal of amino group in a nucleotide base is a metabolic reaction. Hydrolysis of glycosidic bond is an example of metabolism. So guys, from this slide, the only thing which you have to study is all the biochemical reaction taking place inside a living system constitute the metabolism. So important point number 1. Okay, you will get more question from the part related with the enzymes and all. Okay, so guys, we'll learn about this graph, the TCA cycle and all. We will learn in the chapter respiration in plants, which would be taught very soon for you. Okay, which will be taught very soon for you. So the intermediate products of metabolic reactions are referred to as the metabolites. So guys, already when I discussed you the NCERT in the 4th lecture, I told you we missed something, but it was in this slide I used to prepare. I prepared that in this slide. I included it in the form of a slide in this particular session. That is, metabolites are the intermediate product of metabolism. They are of two types, that is primary metabolites and secondary metabolites. Primary metabolites and secondary metabolites. The flow of metabolites in the metabolic pathway has a definite rate and direction like the automobile traffic. You have to understand that the metabolite flow is a dynamic state of the body constituent important. Metabolic flow, it can be asked as in the options and all. Choose the correct one and all. Metabolic flow, metabolite flow is called the dynamic state of the body constituent. Clear everybody? Clear everybody? Yes. So that is actually metabolites. So let's discuss about the two types of metabolites. That is the primary metabolites and the secondary metabolites. Okay. What do you mean by primary metabolites and secondary metabolites? Primary metabolites means they have identifiable function. Joining for the transport lectures and all, or you miss the initial part, so you're continuing or you're planning to join for the class from the new chapter onwards. Yes, that chapter is also important. Okay, so primary metabolites, they have identifiable functions in the physiological processes. Okay, identifiable functions in physiological processes. So the examples of the primary metabolites is amino acid, sugars, nucleic acid, lipids, and vitamin. Yes, everybody. So those metabolites which have an identifiable function in the physiological processes is known as primary metabolites, whereas secondary metabolites means they are not directly involved in the normal growth and development or reproduction. Secondary metabolites means they are not directly involved in the growth and reproduction. This is an important statement. The secondary metabolites are found in the plant, fungal, and bacterial cell. Important, this came as a statement for previous year question. Example we have already discussed, that is alkaloids, pigments, terpenoids, essential oil, toxin, lectins, drugs, etc. So guys, you have to just identify or you have to just understand that those metabolites that have a role in the physiological processes is called primary metabolites, and those doesn't affect the physiological processes is called secondary metabolites. I have told you this particular thing. Okay, I have explained this there. This table is for you to by heart. Okay, I will tell you what all are the previous year question or those questions which you have to expect for a previous year or a question for your NEET exam. That is alkaloids. The most important thing from this entire table is lectin, concavalin A or lectin. Toxin or abrin and rixin, important one. Pigment, alkaloid, that is pigment, carotenoids and anthocyanin. Alkaloids, morphine and codeine, very important guys. Polymeric substances, rubber, gums and cellulose. Drugs are also important, vinblastine and curcumin. Done everybody. Among this, the most repeated and frequently asked question is the lectin, concavalin A, and the drugs vinblastine and curcumin, and the toxin abrin and erixin. Clear? Yes, everybody. So please pay attention towards these three. These three are repeated previous year questions. So this can be again repeated for the examination as well. Okay, everybody. So please note it down. These three is very important. Clear? Let's move on. These are the series of linked in multi-step chemical reactions in metabolism. Okay, you don't have to do in detail about this. You will learn about this in your higher classes. As I have already told you, metabolic pathways are of two types, that is anabolic pathway and catabolic pathway. Anabolic pathway means it is a constructive process, that is small molecules are joined together to form complex molecule. Catabolism, it is a destructive process. What is catabolism? It is that is breakdown of breakdown of complex molecule into simple molecule. Just remember like this, guys. Cat. What is cat? Cat is a small animal, right? So cat is a small, so it is broken down into small molecule. You can use simple chords like this. Okay. It consumes energy, it releases energy. So anabolic processes consumes energy, catabolic processes release energy. Respiration is an example. Respiration, this is very important. Respiration is what all points which I am underlining, you must make sure you got the point very clearly. Okay, everybody listen. Respiration, once again. Okay, respiration is what? An example for a catabolic process. Okay. Okay. Clear, everybody? So catabolic process example is respiration and glycolysis. About glycolysis we will learn in the chapter respiration in plants. And guys, synthesis of proteins from amino acid is an example for an anabolic process. And exactly like that, photosynthesis is also a process that consumes energy. So it is also an example for an anabolic reaction. Once again, I am telling you this, the only thing which you have to by heart over here, guys, anabolism and catabolism. Anabolism means it is a constructive process. Anabolism is a constructive process. Catabolism is a destructive process. Anabolism consumes energy. Catabolism releases energy. Okay, anabolism consumes energy and catabolism releases energy. Guys, anabolism example is photosynthesis. Okay, photosynthesis is an example for an anabolic reaction. And synthesis of amino acid, that is synthesis of proteins from amino acid, is also an example for an anabolic reaction. But catabolic reaction example is glycolysis and respiration. Is it clear? Is it clear? Is it clear, everybody? Done. Let's move with the next part of the chapter. Guys, there is nothing detailed to study. I'll be telling only those things which you have to study. The energy released during the catabolism is stored in the form of chemical bond. Guys, don't worry if you cannot just understand everything. Just whatever points I'm telling you, just by heart those points. That's the important one. Okay, because this chapter, the end part of this chapter is quite difficult for everybody because there is a lot to by heart. Okay, so when needed, this bond energy is utilized for mechanical works. Guys, very important statement. The energy currency of a living system is adenosine triphosphate or ATP. Just one point you have to remember this, only one point you have to remember. The energy currency of a cell, ATP molecule or adenosine triphosphate. Done. MCQ number 1 previous year. From this is a very important slide. You will get a lot of questions from this. Metabolites in different organisms are present in different concentration. Blood concentration of glucose in a normal person is 4.2 to 6.1 millimole per liter. MCQ number 1 from the slide, everybody. Concentration of hormones is measured in nanograms per ml. So blood concentration, the blood concentration of glucose of a normal person is 4.2 to 6.1 millimole per liter. Systems at equilibrium cannot perform work. Number 2, MCQ number 2, or the point which you have to by heart. Systems at equilibrium cannot perform work. As living organisms work continuously, they cannot reach equilibrium. The most repeated question of this part is the point number 3. I am marking it with yet another star marks as well, in order to make sure that this is a very important one. That is, the living state is a non-equilibrium steady state to be able to perform work. Here we have studied that systems at equilibrium cannot perform work, but the living state is a non-equilibrium steady state which is able to perform work. Clear, everybody? So no living state without metabolism. Point number 4. So how many points in this slide we have discussed? The blood concentration of glucose in a normal person is 4.2 to 6.1. Then the living state is a non-equilibrium steady state which is able to perform work. Systems at equilibrium cannot perform work, and there is no living state without metabolism. Give me hearts on the screen, everybody. Give me hearts on the screen, everybody. Yes, come on, everybody. Give me hearts on the screen. Let's move forward with the next part of the chapter. Yes. Next topic is the enzymes. This is a repeated question for your exam as well. Clear, everybody? This can be a question for your exam. What is it? Enzyme. What is the topic? It is the enzyme. Clear, everybody? Let's move on. So guys, enzymes are the biological catalyst. Enzymes are a biological catalyst. Okay, everybody? So just study what all points which I am discussing over here. So today we are going to discuss about the enzymes. Done, everybody? We are going to discuss about the enzymes. So let's discuss one by one. Enzymes are the biological catalyst. Enzymes are the biological catalyst which influences the speed of a biochemical reaction. All enzymes are protein. This is a previous year question. All enzymes are protein, but all proteins are not enzyme. Okay, all enzymes are protein, but all the enzymes are protein, but all proteins are not enzymes. Enzyme action is specific. That is, it acts onto a particular substrate in a particular molecule and all. Okay. Ribozyme. This is again 2019 NEET MCQ. Ribozyme is a nucleic acid. That is RNA molecule that behaves like an enzyme. So that's why we cannot say like all enzymes are protein, but there is an exception towards that. So, sorry, I just twisted it. Sorry. Listen very carefully, everybody. Enzymes is a protein. So all proteins are enzymes. Again made wrong. Really sorry. Let's start once again. Listen very carefully. Come on. Yes. Forget all those things which you have studied in this slide. Forget everything that we have studied in this slide and listen very carefully over here. Enzymes are the biological catalyst. Oh my God. Guys, please give me a yes or no on the chat box. Let's learn this once again, please. Let's learn this once again. The same way which I got twisted, you guys will also get confused. That's the reason. Ready? Just tell me ready on the chat box if you guys are ready for the rest of the session. Yes, everybody. Come on. Ready, everybody? Let's learn this once again in a very detailed and a clear way. Yes, in the examination hall, You will also get twisted in the same way. So please be careful. Okay Fatima. Yes, okay, Calvin and Anantu. So guys, enzymes are a biological catalyst. Point number one, clear? All enzymes are protein. All the enzymes are protein. All the enzymes that we study in the digestive system, then respiration, all the enzymes are protein, but all proteins are not enzyme. You can say all the enzymes. So the major constituent of all the enzyme is the protein. But you cannot say that the enzymes, that is, all proteins are enzymes. You cannot say that. All proteins are not enzyme. Okay, you cannot say every time, you cannot say like all enzymes are protein, but you cannot say like all proteins are enzymes. So make the point very clearly. All enzymes are protein. Once again, I'll remember, make you understand. All enzymes are protein, but all proteins are not enzyme. Enzyme is specific, that is, each has its own substrate. Ribozyme, another previous year question. It is a nucleic acid, guys. An RNA that behaves like an enzyme. An enzyme form a tertiary structure. We have already studied for the biological activity of enzyme. The protein should exhibit a tertiary structure. So those enzymes form a tertiary structure that have certain site called the active site into which the substrate fit. Guys, just remember about a key, lock and key mechanism. Just this, just everybody, just everybody, just look at this diagram. Just look at this diagram. Look at this diagram very carefully. Okay, I just want to tell you something from this diagram. Clear this much. This lock and key mechanism, you just suppose. Okay, this is what I want to tell you, something super important. So listen carefully. Can you all see the zoom diagram? Can you all see the zoom diagram, everybody? Yes. So listen very carefully, everybody. Guys, this is a substrate molecule. This is a substrate molecule. Guys, in your doors and all of your house, you will have a small thing like this, right? And into which you will insert this key. The key will be designed in such a direction or such a manner in which it can directly fit inside this particular lock, right? The key is actually set in such a way that it can actually fix itself inside the lock very easily, right? So exactly like that, a substrate molecule will be there. It may be a protein, carbohydrate, nucleic acid, or whatever. So exactly like that, the biomolecule, the substrate molecule, will also have a shape, and it will have a particular shape in which the enzyme will be designed. Listen everybody carefully. Guys, if this is a particular substrate, you remember, can you all see this? Just imagine this is the substrate molecule. This is the substrate molecule. Then what will be your shape of your then the shape of the enzyme that is actually contributing with, so that this can fit together. Am I right or not? So that this can fit together. This is the enzyme. This is the enzyme and this is the substrate. So they can fit together. Look at the fitting diagram. Everybody listen carefully. Listen carefully, everybody. I will take a blank slide and explain it for you. Everybody listen carefully. Yes? Listen carefully. So guys, this is the shape of my enzyme. Okay, this is the shape of my enzyme, and this is the shape of my substrate molecule. Okay, this is the substrate molecule. Guys, they can fit together, right? Just look, everybody. This is the substrate molecule. And this is my substrate molecule. And this is my substrate molecule. And let me draw my enzyme molecule as well. My enzyme molecule will be having a shape like this. It will be exactly fitting. This is my enzyme molecule. So this mechanism you will call them as lock and key mechanism of enzyme action. Lock and key mechanism of enzyme action. Clear everybody? You don't have to study in detail about this because you will study this in detail in the chapter chemistry. Okay, in chemistry chapter you will learn in detail about this. Okay everybody, this slide is no more important. Okay, there won't be any questions coming from this part. In for the past 33 years no question have been come from this part because there are more important slides than this. So I'm just reading it and going. You don't have to by heart or study all these unnecessary valid information. Okay, so physical changes here, a chemical changes here. You have already learned what is a physical change, what is a chemical change in your lower classes itself. Clear everybody? This can be taught. I'll just tell what is this. You can just study this. That is rate of a chemical reaction. Rate of a chemical reaction means it is the amount of product formed per unit time. Okay, rate of a physical reaction is equal to the amount of product formed per unit time. Just you have to identify that. That's enough. Rate of a reaction is called its velocity and the direction is specified. Clear? Carbonic anhydrase is the fastest enzyme. You will learn in your chapter respiration. That is breathing and exchange of gases of human physiology. You will learn about this carbonic anhydrase. So guys, carbonic anhydrase is the fastest enzyme known, and you have to identify that the carbonic anhydrase is activated with the help of an element that is Sing2 plus ion. Sing2 plus is an element that or metal or an element that is present in the carbonic anhydrase. Clear everybody? You can transfer only 200 molecules of carbon dioxide without the presence of an enzyme. But in the presence of an enzymes like carbonic anhydrase, about 6 lakh molecules of carbon dioxide can be transformed. Just you have to understand carbonic anhydrase is the fastest enzyme and it accelerates the following reaction 10 million times. Okay, it can accelerate 10 million times the reaction. Okay, the glycolysis we will learn in the chapter respiration. Okay, respiration in plants. I will be definitely taking you each and every single point related with the chapter of glycolysis. This entire cycle is for you to by heart. Okay, this entire cycle is there for you to by heart, starting from glucose till the pyruvate. Starting from a 6-carbon compound glucose, you will get a pyruvate. This is the conversion. It is followed by a series of chemical reactions like this. Clear everybody? Glucose C6H12O6 is converted into two molecules of pyruvic acid C4H4O3. Clear everybody? What we have already discussed. This is what we have already discussed. That is a catalytic cycle or the nature of enzyme action. It is a lock and model. Clear? Guys, I told you this is the substrate molecule. This is the substrate molecule. For the substrate molecule, you will have the enzyme would be like this. Or you can take exchange, vice versa also you can take. If this you will take as a substrate, this will be the enzyme. If you take this as a substrate, then this will be the enzyme. It will exchange itself. Clear that? Look, it is fitting together, right everybody? It is fitting together in this particular molecule. Clear? That is actually the enzyme action. Clear? You don't have to by heart all these things. These are unnecessary information given in the textbook. While you're doing, the most important thing which you can do is that I hope all of you will be having an MCQ solving book. So please go to that, do the MCQs from this part after reading the NCERT. Try to do the MCQs from this part. Today we won't be having the NCERT highlight after this. This was the last slide that we see. And how do enzymes speed up a chemical reaction? You know that enzymes are biological catalysts. In chemistry, you have studied that catalyst accelerates a chemical reaction, right? Catalyst speeds up a chemical reaction, right? So we have to understand, just in a brief way, how these catalysts or the enzymes accelerate a chemical reaction. We have to understand, and that we will do in the next class. Okay, just do one thing before your 11 a.m. class of transport in plants, which is going to be the final lecture of the chapter. Take out your NCERT, read the portions before the activation energy of an enzyme, and then you start solving MCQs from this part so that you will be able to know what all points you have to by heart from the NCERT. Done? So that's all for today. Thank you, everybody. See you in the next class with a super amazing lecture. Bye bye, everyone. Don't forget to attend the session that is at 11, Transport in Plants final lecture, and also don't forget to attend the examination at 7. Done? Done, done, done. Bye bye, everybody.